Yesterday on Monday, I gave you something to talk about with those around you, and it was basically this: if you were asked to put together the absolute best phylogeny possible, how would you do it? For example, what data would you use? How many taxa would you try to connect? Okay, what different strategies would you use as you build that table? So we came to the conclusion, we came to some conclusions. One of those is that molecular data are more objective than morphological data, right? What are molecular data again? What are those data? Give me an example of molecular data. Yeah, Levi.
Cell interactions.
Okay, what do you mean by that, David?
RNA.
RNA, but what about RNA and DNA? The sequences, okay? The DNA sequence, so the sequence of bases making up a particular gene, or the sequence of bases making up the RNA transcript of that gene, or the sequence of amino acids in the protein, those are molecular data. Why are those more objective than morphological data? Yeah, Chris.
Morphological data might vary depending on the environment or the type of situation.
Especially when you have analogous features, right? Where you have two similar structures that develop because of similar environmental need, but they develop completely different, right? The organisms don't share recent ancestry. And then so you get something complicated with morphological data. On top of that, you have to decide what is more difficult to accomplish. You know, developing this morphological feature twice or developing this one twice, right? Which one's more difficult to accomplish? With molecular data, you don't really have to worry about that, because anytime you would have a question of like what mutation is more difficult to accomplish, we have data to support that. We know which types of mutations are most common. We know where mutations are most common. Okay, we have those data. All right? But we talked about some of the complications for using molecular data only is that it assumes a molecular clock, and we know that not every organism has the same mutation rate. We know not every gene has the same mutation rate, and we know actually that there's some amount of variation. On top of that, we have this question here: how does horizontal gene transfer complicate molecular phylogenies even more? Okay, HDT stands for horizontal gene transfer. And so horizontal gene transfer happens when DNA from one organism ends up in the genome of another organism. So within the same generation. This isn't passing it off to your offspring. That's what we call vertical gene transfer. This is horizontal gene transfer. This would be something akin to, you know, I have blue eyes, you want blue eyes, so I give you the genetic material necessary for your eye color to change. Okay? This is the idea of horizontal, sometimes referred to as lateral gene transfer. What this does, yeah, Faith?
Would that, an example of that be like how they're trying to, basically you can give your child like...
Yeah, so it's a little bit different than designer babies, but yes, but in that, I mean, that's basically what you're doing, is you're changing the gene in the embryo to something you want, a different version of that gene. And there just an article came out, not yesterday, I think it was 2 days ago, on Monday, maybe it was last week, where for the first time, Chinese scientists changed the genome of two babies that were born to parents that both have HIV, that both have AIDS. And so they changed a particular gene in the embryos to make them resistant to the virus. And so now they are developing in utero with a version of the gene that should lead to increased resistance to actually succumbing to the virus and developing AIDS. And so that is horizontal gene transfer, but it's different than it just happening mechanistically. There you're actually doing some genetic manipulation, and there are various tools by which you can use to do that. Micah?
How does it just happen mechanistically?
That's a wonderful question. We'll talk about that in just a moment. Yeah. Although what's a more fascinating question is, how do you make it happen? right? So they have gene guns. I don't know if you know that, where you can fire a piece of DNA into the cell. You don't do that with a developing embryo, right? Like you may do that into, 5,000 seeds of a plant, and you'll fire a gene into them and then hope that some of the seeds survive and actually develop, right? But you don't do that with embryos. And so what they do is they use viruses, and they use material from bacteria to actually they inject it into the embryo, and it reverses the DNA back into the human genome. Because there are a number of viruses that do that. Remember, we talked about that in chapter 21, that there are some viruses that they have DNA, and so they kind of insert themselves into the host DNA, and so you use some of that machinery to make that actually work. Anyways, this is another story for another time. So, but what horizontal gene transfer makes possible is that even some of the molecular similarities could be analogous and not homologous. So my brother and I could both have blue eyes. We don't. But my brother and I could both have blue eyes, and you would assume that because we're brothers, that we both inherited those blue eyes from our parents, right? That would make sense. That would be a reasonable thing to assume. But because of horizontal gene transfer, it's actually possible that either of us could have gotten that gene from another individual and not from our parents. Okay? And so what this does is it makes it possible for molecular similarities to not be because of shared ancestry. So horizontal gene transfer is incredibly common in prokaryotes. Okay? Incredibly common in bacteria, archaeans, so much so that some would argue that we can't even talk about separate species of bacteria. Since they all share genetic information or are capable of sharing genetic information, it's irrelevant to refer to them as separate species, and it might be more accurate to just refer to them as a single species with many different morphs. Now, we are starting to find more evidence of this in eukaryotes, though, and I'll give you some examples of it in eukaryotes and then talk about some potential mechanisms. All right. So aphids are insects, and they eat red fungi and they turn red. Okay? And so it's like, oh, cool, it's like flamingos, right? I don't know if you know this or not, but flamingos are not born pink. They're born like grayish white. And then they eat a lot of the small crustaceans in the water that have concentrated pigments from algae, and then it turns their feathers pink. And you're like, oh, cool, that's what's happening in the aphids. But it's not. The aphids actually now have the same gene that the fungi use to produce that red pigment. Aphids now have and produce that pigment. And so then if you knock out that gene, they still eat the red fungi, but they're not red, because they're no longer making that red pigment. And so it seems that the gene for making that red pigment has moved laterally from the fungi to the aphids. This is fun. Transposable elements are mobile genetic elements, and usually they just jump around a single genome. Like a piece might jump from chromosome 1 to chromosome 5. Or from chromosome 5 to chromosome 21, right? They're just mobile genetic elements. But we're starting to find evidence that these transposable elements can jump from one organism to another. And in this case, from rice to another grain to millet. And so here's one possible mechanism. One is parasites. That there are animals, we've talked about some of these already, that live in multiple different hosts. And the idea is that maybe when they are in a host, that they take part of that host with them when they go to the next host. And that part of the host that they take with them may be some of that host DNA. And then so they move from, let's say we're dealing with, we could do anything, one of our filarial worms. Okay, like Wuchereria bancrofti that causes elephantiasis. And as it moves from a mosquito to a human, that it has a potential to take mosquito DNA and actually insert it into its human host. We know that viruses have the potential to do this because viruses will insert their own DNA into the host genome, and then when it pops out to build a new virus, it pulls some of that host DNA with it. And so there are some stink bug species that have the exact gene in proper form to make black widow toxin. Now, they don't have a means of delivering it like black widows do, but they now have a gene to produce black widow toxin that builds up in some of their tissues without having any means to inject it like a black widow spider does. And the best way to explain that, and to answer your question, Micah, the mechanism is that if the same virus infects both these stink bug species and black widow spiders, that provided the mechanism by which you can pull some DNA out of the spider genome and insert it into a different host. Now, you're limited with viruses because viruses are small. They have very small genomes. But it does provide a suitable mechanism. And so here are the aphids that are expressing the gene, the fungal gene, to produce the red pigment. And here are the same aphids where that gene has been knocked out. So they still have that gene, but they can no longer express it. They're still eating the red fungi, but they are not red because they are no longer expressing and forming that pigment. Yeah, Allison.
So can horizontally transferred genes then be passed down to the next?
Oh, absolutely. Yep, once they're in your genome, as long as they are in the germline, right? So if the horizontal gene transfer takes place in a non-germ cell that's not going to develop into a gamete, then they can't be passed on. But like if you change the zygote, the original cell, and you modify it, then every cell that descends from that is going to have that modified version of the gene. All right? Does that make sense? Okay, we're going to take a lecture break right now, and what we're going to do is we're going to look at one question that I put together to just kind of sum everything up, because this was supposed to be our second to last chapter that we discuss, and so these questions all are focused on summing it up. I'll make sure that your version of these slides have all these questions. We're going to talk about one of them during our lecture break, and then what I want you to do is be prepared to answer any of them on our exam a week from Friday. All right? By the way, by Friday, I'll get you a study guide posted for that exam. All right. Okay, so first question, this is the one I want you to deal with right now during our lecture break, is taking into account everything we've learned in the various groups in fungi, in plants, in protists, in bacteria, in viruses, in animals, assuming that what the Bible teaches about a global flood is true, that there was in fact a global flood, and it contributes a significant amount of the material, a significant amount of the rock material where fossils are found. Where would we have these boundaries? Okay, so if you're thinking back to, we talked about Paleozoic, Mesozoic, Cenozoic eras, right? It's been a while, but we've talked about these. Where would you likely mark the beginning of the flood and why? And where would you likely mark the end of the flood and why, based on what we've learned so far? We'll take 3 minutes. 3 minutes, work with those around you, come up with an idea of where the flood deposits begin and an idea of where the flood deposits end.
I'm like, yeah. Okay, yeah. That makes sense.
Because like people say that all the animals every yeah.
And the only way that can happen is
That's like when they're it's because not till
It's actually very rare for a body that died up on the surface to get into the fossil record. It has to be buried by something, and so that's why I think Cambrian is supposed to be
yeah, that's what
whenever there's like a tsunami or a flood somewhere and we're all alive well, even though it's like dirt, it's in the
Yeah.
and so that's like, it's just that shit. We'll get into say there was a flood's gone, and you walk like a different, and there's like this layer of dirt on the ground, and it's like water being kind of half. If that flood had gone on for a long,
Like for a year.
that level of sediment would have built up. So that's why
Okay, so where are the flood boundaries? So we got a good idea of when it would have started. I would assume it ended right here because that's what they say, like, if that made, yeah, you know, because they think all this took, I don't know, 500 million years to happen, but we know it happened within 1 year, and so, like, we could just condense all that down into 1 year, so I'd assume it would end almost to present time, because that's what happens.
Yeah, right.
So,
Flood was like 40 days, wasn't it?
it rained for 40 days, the flood lasted for like a year.
Yeah.
He was
in there for a year? Poor guy.
It's a long time to spend with your kids. I don't envy him.
it rained for 40 days and 40 days and 40 nights.
All right.
That's just the longest time. There was
So,
just a time after.
Here's the geologic record, okay, or the geologic column. Everything before this is considered to be Precambrian deposits. This is somewhere around 540 million years ago. There are different dates, but that's probably the most current. And so then we have Paleozoic era, Mesozoic era, Cenozoic era, and then here are all of our periods. Okay? So here's our geologic record. And in order to answer this question, you really have to answer the question, what are the key events in the fossil record? What are the key events in the fossil record, Amanda?
I would say the Cambrian explosion. It's where the most fossils were found that were in
The greatest diversity of fossils.
yeah, the greatest diversity of fossils, and that makes me think that the Cambrian explosion is most likely where the flood at least started, because it had like the massive sedentary deposits that were shifted around with the flood.
Yeah, so every single animal phylum shows up here, including Chordata, or Chordata, our phylum. Now, vertebrates don't show up here. Vertebrates don't show up until a little bit later, more like the Ordovician, you start finding fishes, okay? And then up here, Devonian, you start finding terrestrial vertebrates, okay? But all animal phyla show up right here in the fossil record. That's not the same for plants. Some plant phyla have kind of a different story, but it's definitely the case for animals, okay? That all of our animal phyla show up right here. And as far as the number of phyla are concerned, this is where you have the greatest amount of fossil diversity, is right here in the Cambrian. It's where you have the greatest amount of diversity of phyla. So you're suggesting that this would mark the beginning of flood deposits, because that's the best way to explain the sudden appearance of an enormous amount of animal diversity. Levi.
I was just gonna ask if it was kind of like a gradual thing, like you kind of build up to having everything, or if it's like,
I mean, there are some animal fossils in Precambrian deposits, but it's not subtle. It's like you've got a few different fossils, and then it's just boom. I mean, it really is like an explosion. There's a reason why we call it the Cambrian explosion. Okay, it is a very, very significant event. Very significant event. So either you're looking at something that historically is something very significant that happened on Earth, or alternatively, you're looking at basically the beginning of the flood, or the beginning of the burial. Not necessarily the beginning of the flood, but the beginning of the burial events. All right? So we have an idea of where it begins. Anybody want to provide an alternative for when it might begin? So we have one option. Begins right here, along with the boom. Man, that was clever. That's clever. So we talked about this before. We talked about it in our chapter on bacteria, that there are a number of fossil bacterial biomatrices that are found. We call them stromatolites. We find some of these in these deposits. So some have proposed a bit of a later start date, but this is by far the most common proposal for the start of flood deposits. So then everything before this would represent the Earth prior to that flood event. Okay? All right. Now what about for where it ends? We need some key events. Yeah, Micah.
So I was saying that there was probably a higher concentration of land animals being buried on land after the flood.
Okay.
So you'd see this mixture of the explosion.
Yeah.
And you'd see more land animals being buried on land and no marine animals.
Yeah, so you get, now what's interesting, so you'd look for where do we get to a point where the number of land species outweigh or outnumber would probably be a better indication, outnumber the marine? You won't find that anywhere in the fossil record. Oh, actually, that's not true. I lied. I lied to you. It was inadvertent. But I lied to you. You do actually... It's murky, but you find something. Wow. You find something around here. Land creatures outnumber marine. Somewhere around there. Mia.
Do you think you could start looking where the vascular plants started?
Sure.
So that would be. I have in my notes that it's around
So,
I'm not sure if that's right.
yeah, so that's when ferns would show up, which are vascular but still seedless.
Okay.
And ferns are definitely show up about right here. So we've got bryophytes that show up first and show up here, and then we have ferns that show up, so we've got some vascular plants. Amanda?
I think another possibility is not necessarily when the land creatures outnumber the marine, but there's a significant increase in the number of land fossils of land creatures starting to appear among the marine.
Okay.
So just even an increase.
Okay. Yeah, and so that actually starts to happen right here. Large increase in land creatures. Man, I like that. Land creatures. I was thinking of writing land dwelling forms, but land creatures just sounds so much better. Yeah, Tara.
I wouldn't know what time period, but I guess just when the amount of fossils being found matches the pre-Cambrian explosion, the pre-Cambrian period.
Oh, yeah, so the density?
The end of when something catastrophic would be laying down that amount of fossils.
Yeah, there's no point where the density matches this, but there are multiple ways to explain this. These rocks are buried very deep and experience a lot more activity, geological activity, than rocks that aren't buried as deep, and so the fossils tend to disappear, right? As you subject rocks to enormous geological activity, we do see a drop in fossil density. We'll just say that. And now it doesn't get down to what you find in Precambrian deposits, but we certainly find a drop in fossil density. How about when we start finding modern forms, right? That I think would give us a good clue as to where the flood might have ended, because now what we're going to have is a result of what survived the flood and then diversified from there. And so we start getting modern forms here. And so like I said, we represent every animal phylum here, but when you're talking about like modern forms of vertebrates, very few of them go back before here, especially for mammals, birds, reptiles. Very few of them go back behind here. Some of them will start to come here. Some modern forms. But most of them don't start coming until here. All right? So we have all these different pieces. Any other significant events? So there are mass extinctions, and I'll put these in a question mark, because if this is actually being laid down during the flood, you're not looking at a mass extinction event. You're really just looking at a transition and what forms were buried and when. So there's a mass extinction here, and there's a mass extinction here. There are others, but these are the two biggest. Okay, and so when you look at Paleozoic forms versus Mesozoic forms, completely different. Because we had either a mass extinction or a massive transition in materials laid down during a global flood. Same thing when you look at Mesozoic versus Cenozoic. Major difference in your major, especially your land-dwelling forms. Yeah, Allison.
How does the human fossil record fit into this?
Oh, yeah. So humans. I have to remember where the Paleogene-Neogene break is, but humans are like here. Very, very small amount of the overall fossil record.
Is that because maybe they tried to get to safety, or did you not know what?
Yeah, I don't know. I mean, there are proposals for. I mean, you really have the problem not just with humans, but why don't we find other forms earlier on in the fossil record, right? If they existed during the flood or before the flood, why don't we find them until, after the flood deposits? So we really have that issue with a number of forms. I mean, some of it you could try to explain it as your ability to get away from where you're going to have the most incredible amount of activity. Makes it very unlikely that you would be buried, but more likely that you would just drown and then be eaten by something before you had a chance to be buried. So I don't know. I kind of like the idea. Some people will propose that God said he was going to erase all evidence of humanity, right? Other than the remnant that he chose. So, but I don't know. We have that issue with a number of forms, not just with humans. So the most common proposal for the end of the flood deposits is right here, at what's called the KT boundary. And it seems weird to call this the KT boundary when this is Cretaceous. That doesn't start with a K, although it does in German, I think. And then this is Paleogene. That certainly doesn't start with the T, but there are other names for it. But anyways, we call this the KT boundary, and that's where all dinosaurs other than birds disappear. Because phylogenetically, birds are dinosaurs, so other than birds, all dinosaurs disappear at this boundary. Okay, most flood models begin here at the Cambrian explosion and end here at the end of the Cretaceous, transitioning into the Cenozoic. Okay, so this is the most common place to put the end of flood. There are some flood models that take it more recent because in conventional geologic time, this is about 65 million years ago. Okay? Which would mean that we would lay down about 500 million years of rock in less than a year, and then we would lay down 65 million years worth of rock in the 4,000 or so years that have passed since the flood. All right. Okay. Yeah, Amanda.
It's possible that because if I remember in the Bible is that prior to the flood, humanity like had a much longer lifespan.
Yes. Mm-hmm.
So it's possible that that it could have also been a possible reason why it looks like in terms of the fossil record that the flood was over a long period.
Sure.
Of time.
Yeah, so, population geneticists have gone and tried to determine if there are no gaps in any genealogies, and there's about 2,000 years or so that to pass between creation and the beginning of the flood, how big would the human population be? And most estimates are somewhere around 1 billion. That with current growth rates, and you look at that there'd be about 1 billion people on Earth at the time of the flood. That's interesting to have 1 billion people and have no record of it. So, although you'd have way more than 1 billion angiosperms based on modern angiosperm distributions, and there are no angiosperm no fruiting plants until you get to here either. And regardless of your view of origins, angiosperms had to have appeared way before they actually appear in the fossil record. So, yeah, Micah.
What do we do with this huge gap of time?
Which huge gap of time?
Well, you said the 500 million years between.
How are we coming up with those numbers? How do we deal with that?
Yeah.
Well, so those numbers are based on primarily radiometric dating of the rock. And so the way you would typically deal with that is to just say there's something wrong with our dating techniques. Okay? Now, when you point out there are 3 big assumptions with radiometric dating, there is that the rate of decay is constant, the amount of parent material that ends up in a living form or a living rock is always the same. So, like when you have a rock formed 500 million years ago, the same type of rock that forms today has the same amount of parent material. And then the other one is that there's no contamination. That material can't leave the rock. All it can do is decay into something else. 2 of those assumptions we know are wrong.
So could you say that they're looking for what they want to find?
Maybe, but what's really interesting is when you determine a date of a rock, and say it dates to the Silurian, and then you have a phylogeny and you propose based on the molecular information of when that organism would have had to exist just based on your mutation rate, and you get the same date. So when your theoretical date matches your measured date, then you're really starting to deal with something that's intriguing. Now what's interesting, there are issues with these. So they've dated rock that formed in Mount St. Helens eruption in the 1980s, and it'll provide dates of much, much longer than 30 years. And so it gets a little bit interesting, especially if, anyways, we'll talk, I don't know if you'll talk about radiometric dating a whole lot in Gen Chem. We'll talk a little bit more about it in cell biology. But anyways, when you get outside of like a half-life, the accuracy really breaks down. And for most of these techniques, the half-life is 500 million years or 1 billion years. right? And so if these rocks are only thousands of years old, you're way outside of the half-life, and the accuracy is not going to be reliable.
So have Christians
Proposed an alternative?
alternatives?
Yeah, so, gosh, why can't... Dr. Andrew Snelling, he's the director of research for Answers in Genesis. He writes more about radiometric dating than anybody that I know. So you can read some of his, but yeah, he basically points out mainly the two issues in the assumptions, that we know that the amount of parent to daughter ratio is not the same at all times, that sometimes when rock forms it's this amount, sometimes when a rock forms it's this amount, and we know that there's movement of materials in and out of rock. So he attacks it mostly that way, and what he'll say is that we're still seeing relative dates. So rocks here are certainly older than rocks here, but that these are not absolute dates. Amanda.
Could it be possible that the reason why we have the timeline of the fossil record the way it is because the flood shifted certain sediments to like from one area to another?
Oh, absolutely.
So that could have probably affected.
Yeah, and you have to realize sediment's only going to accumulate in a basin, right? So you have to have like a sink for sediment to accumulate. That's where it's going to accumulate. So sediment's not going to accumulate in some places just because water is not going to flow and stop flowing at a particular place. So yeah, sediment is carried all over the place.
And so that probably could have like caused.
It complicates the picture a lot, for sure. And we have no idea what a global flood would look like and what it would be capable of doing, right? When you try to extrapolate from, say, like when the levee broke in New Orleans during Hurricane Katrina, right? And the amount of just destruction that happened, I mean, that is a tiny event compared to a global flood, right? And so to know in terms of the amount of geological activity that would accompany it, I mean, it's just hard to know. All right? So that's the end of chapter 20. We're going to transition from chapter 20 back to chapter 18. But before we do, I just want you to look at these. I'm going to make sure that your version of these slides have these questions. Or I could just ask, do your versions of these slides have these questions already?
No.
No? Okay. So I will give you these questions. And I want you to know these are going to be very, very important questions to be able to answer. All right? Okay. All right. Chapter 18. We're going to just do the first framing question, and we will save the rest for future class. So how would natural selection function in a population of plants? So in chapter 19, we talked about the mechanisms of evolution, right? Right?
Yes.
Okay, and there were 5 mechanisms of evolution. And what are those 5 mechanisms? So natural selection is one of them. Okay? Mutations, gene flow, genetic drift, and sexual selection, which you could argue is actually a form of natural selection if you want. And so in chapter 18, which is interesting because chapter 18 comes before chapter 19, but in chapter 18 it's like, okay, well now let's apply these concepts to populations, and it applies concepts you haven't learned yet, which is fun, which is why I reversed it, but then I got all mixed up. So remember that natural selection is survival of the fittest, and so the most fit individuals are going to leave the biggest impact on the gene pool. And why is that? What does it mean to be fit? Levi. Yeah, fitness is entirely a measurement of reproductive success. Okay? And if you are successful, if you are fit and you have a high reproductive success, you are not just producing a lot of offspring, but you are producing offspring that are viable and are going to produce their own offspring, right? Because if you make, I mean, from a biological perspective, if you produce 30 offspring that are all sterile, it's equivalent to producing 0 offspring. That make sense? If you produce 30 offspring that are all sterile, it's equivalent biologically to producing 0 offspring. Okay? So that's a good piece of information to keep in mind. Now, if you were to take several seeds of the same plant and spread it in your garden, you have now created a population. Okay? So let's say you take, I don't know, broccoli, because it's delicious, and you plant it in your garden, you now have a population of broccoli plants, which I don't know if you know this, but broccoli grows from mustard. It's derived from wild mustard, as is cauliflower and collard greens, and I think Brussels sprouts, and mustard, of course, and broccoli rabe, all sorts of different, kale. They all come from the same plant. Anyways, this is another story for another time. So we have a relatively isolated population, right? It is physically possible that an insect is going to come and take some pollen from your broccoli plant and deposit it in the flower of a different, we'll call it a mustard plant because it's not a broccoli plant, okay? Right, that is possible. But if you've got a garden, you're relatively reproductively isolated. You probably will have some gene flow, but not a great. Now, the most fit individuals are going to produce the best seeds. Okay? If a plant has a high fitness, it is going to produce the seeds that are going to have the greatest chance of producing viable offspring. So they're probably going to grow the fastest. They're going to reach reproductive age most quickly. And because a number of plants self-fertilize, the fit individuals are going to continue to pass on their genetic material to the next generation. Now, in the event that one of our fit individuals pollinates another fit individual, so you cross-fertilize, it's possible that you could in the future have individuals that have even higher fitness than the previous generation. Now, this will work the same way in animals that are capable of self-fertilizing. But any animals that aren't capable of self-fertilization, or any plants that aren't capable of self-fertilization, or any other diploid organism that isn't capable of self-fertilization and relies entirely on sexual reproduction, your natural selection is going to always basically be this form. That you're going to take one individual, you're going to cross-fertilize with another individual, and you have the potential to create unique copies. Unique versions, probably a better way to state this. And so I put this question on there and wanted to talk about it to get you to think in the context of what's actually happening as natural selection happens. And so then as we start talking about, okay, how can this produce two different species, we're all on the same page. All right? We don't have time to get through the next question. The next question's a heavy one. So we're going to go ahead and end class here. It's about 12:33, which is 7 minutes before our class is scheduled to end. So if you want to hang out and spend those 7 minutes asking questions, feel free to do so. If you don't,